When a manufactured pharmaceutical leaves the production line, it begins a slow, inevitable process of thermodynamic decay. The central challenge of drug manufacturing is proving mathematically that a dosage form will remain chemically and therapeutically viable months or even years into the future. We define this property strictly as pharmaceutical stability. It is the verified ability of a drug substance or product to maintain its exact physical, chemical, macrobiological, and therapeutic properties within predetermined specifications over a set duration of time. Three primary environmental stressors drive the degradation of these active compounds: ambient temperature, relative humidity, and direct exposure to light. By systematically isolating and measuring the impact of these stressors in a controlled laboratory, manufacturers establish a precise expiry date, dictate required storage conditions, and verify the physical integrity of the product's packaging system. Stability studies translate the physical realities of the environment into legally defensible shelf life determinations. They form the empirical, scientific backbone of pharmaceutical quality assurance. Establishing a multi-year shelf life before a product actually sits on a shelf for several years requires a combination of real-time observation and predictive mathematical models to forecast the degradation trajectory. This table outlines core methodologies. The baseline long-term stability study observes the drug at 25 degrees Celsius and 60% humidity. Accelerated studies subject the product to severe stress at 40 degrees and 75% humidity. Failures trigger intermediate studies at 30 degrees and 65%. While temperature and moisture address atmospheric stressors, light exposure represents a separate degradation pathway that requires a dedicated photostability protocol. In this test, the product is subjected to 1.2 million lux hours of light to accurately map photochemical decay. Finally, formulations are subjected to freeze-thaw cycling. This violently swings the product between -20 and +25 degrees Celsius to test physical resilience against crystallization. Together, this multi-tiered battery of extreme and standard tests creates a highly comprehensive predictive model. It anticipates the exact failure point of a given drug under virtually any physical stress. Once the product is exposed to these atmospheric stressors, attention shifts to the exact scientific parameters measured inside the lab. The evaluation metrics are grouped into 4 strict categories: physical attributes like appearance and viscosity, chemical metrics, and microbiological sterility, and functional performance like dissolution rate. The chemical markers carry the heaviest analytical weight. Scientists specifically track the gradual loss of drug assay, meaning the sheer drop in active potency, and meticulously monitor the emergence of dangerous, toxic degradation impurities over time. This chart visualizes the standardized chronological testing schedule required to capture that chemical decay. Regulatory protocols mandate data collection at explicit intervals: month 0, 3, 6, 9, 12, 18, and 24. Testing at these exact intervals extracts the statistical trend lines. Regulatory bodies rely exclusively on these lines to officially assign and legally defend the final commercial shelf life of the product. A drug stored in the temperate climate of Europe faces baseline atmospheric variables that differ drastically from the exact same formulation sitting in a warehouse in tropical Southeast Asia. To solve this massive geographical discrepancy, the international regulatory community standardized the globe into distinct climatic zones, as shown on this map. We can contrast the mild, temperate conditions of Zone 1 and Zone 2, regions like the United States and Japan, against the harsh, extreme heat and humidity characteristic of Zones 3 and 4. For instance, a product intended for Zone 4B, representing very hot and humid environments, must pass rigorous, long-term testing at a constant 30 degrees Celsius and 75% relative humidity just to establish its baseline. This structured climatic zone framework allows manufacturers to generate a singular, centralized scientific dataset capable of satisfying international safety requirements across wildly heterogeneous global environments. The specific study designs, extraction intervals, and climatic mapping systems we have discussed are not industry suggestions. They are legally enforced through a strict international harmonization matrix. The foundational guideline is ICH Q1AR2. It dictates the overarching general stability testing mandates required for any new drug substance or product attempting to enter the global market. This master framework is supported by specialized sub-guidelines. Q1b regulates photostability, Q1c governs novel dosage forms, Q1d establishes matrixing rules, and Q1e outlines mathematical data evaluation. These centralized ICH frameworks are further augmented by specific regional enforcement documents, notably the World Health Organization's TRS 953 and localized FDA guidance protocols for the U.S. market. A new compound could boast flawless clinical efficacy in human trials, but if the stability data fails to map rigorously to this exact ICH matrix, regulatory authorities will reject the product dossier entirely. We can synthesize these concepts by calculating the commercial label for a new parenteral product, an injectable liquid formulation housed in an amber glass vial. First, we review the long-term empirical data. The formulation successfully maintained its chemical assay at 25 degrees Celsius and 60% relative humidity for a full 24 months. Next, we analyze the accelerated stress data. The batch passed the 6-month predictive test at 40 degrees Celsius and 75% humidity, without generating any out-of-specification toxic impurities. However, the photostability data introduces a failing variable. When subjected to the standard 1.2 million lux hours, the liquid shows unacceptable light-induced degradation, despite passing all temperature benchmarks. Those 3 distinct data points directly dictate the final legal text printed on the packaging. Assign a 24-month shelf life. Store below 25 degrees Celsius. Protect from light. Stability studies provide the uncompromising scientific foundation of pharmaceutical quality assurance. This empirical testing matrix provides the predictive framework guiding manufacturing and logistics, ensuring the drug remains potent and safe until administration. To master the mechanisms of pharmaceutical manufacturing, subscribe to the Pharma Training channel for more highly rigorous GMP, R&D, and quality affairs training content.